I have primarily discussed genome modification in E. coli and to some degree in yeast. The basic mechanisms of genome manipulation are the same regardless of the host chosen, but not all organisms are equally amenable to the various techniques. There are many organisms that have no methods of deliberate genetic manipulation, but many organisms have at least one method. Many bacteria simply can't be transformed. You just can't pop a plasmid into these organisms. In these cases, Plan B is to use broad host range replicons on double-stranded circular DNAs and deliver these via RP4-based conjugation. If there are no known replicons that work in the organism, you can also deliver a transposon via RP4 conjugation, and the DNA will hop randomly into the genome where it can replicate. People often develop species-specific tools for particular organisms of interest. Often this is done by tweaking phage native to the organism or through the construction of shuttle vectors. Shuttle vectors are plasmid DNAs that contain both an E. coli origin of replication and one that works in the recipient cell. There are at least 3 commonly used model yeasts, S. cerevisiae, S. pombe, and P. pastoris, but also many industrially used non-model strains exist. For example, the S. cerevisiae used to make beer, wine, or biofuel is genetically quite different from the laboratory strains. The origins of replication one uses in yeast are different than those in E. coli, but there are yeast plasmids and they are similarly circular double-stranded molecules. S. cerevisiae is awesome at homologous recombinations, so much so that going straight into the genome with a DNA is as common as episomal cloning and similarly efficient. Many yeast have diploid genomes and can be induced to be haploid or diploid. There are many tricks you can do with mating yeast together that cannot be done in the haploid bacterial genomes. It is rare to employ viruses and transposons in yeast. These things are just not part of yeast natural biology. They don't tend to be infected by phages or viruses like bacteria and animal cells are. Many animal-derived cell lines, including mammalian and even humans, are amenable to some form of genetic manipulation. Many human cell lines can be transiently transfected with double-stranded DNAs, which are usually linearized, and they'll last in the cell for a few days. Most experiments are done this way because it's the easiest and most robust procedure. There are episomal replicons like Epstein-Barr-derived DNAs, but that isn't a common thing to do in mammalian cells. What is common is to produce stable cell lines either by random integration of transfected DNAs or by use of randomly integrating viruses such as retrotransposons. There are also Phi-C31 at-site and Cre-based integration strategies for site-specific integration of DNAs into the genome. Targeted genome manipulations are done by homologous recombination. However, this process is greatly enhanced through induction of double-stranded breaks near the site of recombination. These cuts can be generated with zinc finger nucleases, tau nucleases, or more recently using CRISPR. If done in embryonic stem cell lines, stable transgenics can develop back into adult animals. This capability leads to the ability to generate clonal populations of animals. In addition to manipulating DNA in animal cells, most animal cells also do siRNA, and thus gene expression can be manipulated epigenetically. Different animals and mammals differ greatly in the efficiency of these various procedures, and even different cell lines from a single organism may vary greatly in their ability to be modified.